Today, I'm going to introduce the article A Scalable Pipeline for Designing Reconfigurable Organisms by Biotechnology's Journal Club, made by me, Loredana Parisi, Michela Ciocca, and Giorgia Nicolini. So, Loredana, please, you can start.
Okay, thanks, Camilla. And good morning to everybody. And today I will show you this interesting article that we choose it because it's innovative, mostly for its future application. Until today, several methods underway to design and build bespoke living systems that have, of course, different disadvantages. And for example, we have single cell-based methods, which is not scalable to rational control of multicellular shape and behavior. Another method is based on synthetic organoids that is shown in this cartoon, which have a limited control over their structure and function. And finally, we have most recent method that is a bioengineering use of 3D scaffolds, which has the inability to predict behavioral impact or arbitrary biological construction. So it doesn't allow researchers to generate new form of organisms. And in particular, this group proposed a scalable approach for designing living system in silico using an evolutionary algorithm. And they show how the evolved design can be rapidly manufactured using a cell-based construction toolkit. And now we can see how is the approach that they use. And in particular, the approach, in this case that they used, is to give an input to a scalable pipeline. In fact, we have biological building blocks that is shown in the first graph that will correspond to the cells and in the desired behavior of the future organism. In fact, at the end, design that fitting efficiently with the input will be our living system, which embody the desired behavior as the output. And the pipeline is a cycling sequence of generators and filters. In fact, in this case, after giving desired inputs, the algorithm that represents the generator combines the biological building block together to realize a population of random design, as you can see here. And the population is evaluated throughout filters that delete less performant design and overwriting them by more performant design in this case. But now we can see how these algorithms work. So, in fact, we can compare this algorithm to Darwin theory. In fact, principally we are all different because of our phenotype. And for example, this is basically is the reason for different color of your hair, for example. And the phenotype, as you can see here, represents the results of your genotype, formed by all of your genes. In fact, another important point is the fact that the phenotype is the result of your reproduction, of the reproduction of two generator organisms. And in this case, in this process, the genome is edited by crossing over endpoint mutation that generate a new organism. And in fact, so as Darwin told us, species in this case were selected by speciation and natural selection to create a more suitable organism that can survive. So in this case, the algorithm that they used is called CCPN. That means composition pattern producing networks. And in particular, CCPNs are algorithms that through mathematical and Gaussian function allow to stimulate what is the complexity of an organism in nature in a computer algorithm. In fact, they create a network with which it is allowed to analyze all of the mutation of the population. And in this case, in these images, we can see a mathematical way to represent the organism. In fact, in a mathematical way, the organism is represented by a weight matrix formed by voxel, in this case, positioned in the space, and the position is described by Cartesian coordinates x, y, and z that is shown in this image, and that we compared with the genes in conclusion. So the combination of the genes is described by function that in this case we call it genotype, and the final position of the voxel represents the phenotype of the weight matrix. So in fact, this algorithm can be mix the weight matrix and can insert mutation as in the biological reproduction. In fact, you can see here in the image that we can have a single point crossover, 2-point crossover, or point mutation, and so on, that generate a new organism. But an important point at this time is that each matrix has an historical marking, a value that represents the number of acquired mutation. And this historical marking is useful to divide the organism into growing generation. In fact, we can have the first, second, or third generation, for example. And each generation will be subjected to speciation, and so only better design became suitable for the in vivo building, as you can see here. Okay, here in this image, you can see a summary of the process that starts principally with the two inputs. In fact, the first input is represented by two types of building blocks, and we have an inert and a contractile part. And the contractile one is necessary for the second input, represented by the goal, which is the motility of the organism. And in this, at this point, input pass throughout the algorithm, and only the better design survive and are realized in vivo, as you can see in the last, in this part of the image. And now we can see what are the design selection criteria. In fact, we have locomotion, robustness, and buildability. And first of all, locomotion, which allow passage of design that have the ability to move faster than others. Second, we have robustness, which only allow passage of design that sustain the desired behavior in the face of noise. And finally, we have buildability, which remove design that are not suitable for the current build method or are likely to scale to more complex tasks in the future deployment. And to satisfy the buildability filter, that is the first filter, they need organisms that answer three characteristics. In fact, first of all, they need contiguous tissue region because the current build method layers tissue types sequentially, one on top of the other, with which layers filling the X and Y plane. And the second filter, the second point is that they need also stable geometry. In fact, they know that gaps of 100 microns or larger represent the smallest concavity which was stable at the time of shaping and occurs at the last 5 successive days of development. So at this point, if the organism shape include gaps, they must be greater or equal of 100 microns. And finally, design should be 50% passive tissue for energy efficiency and to scale to more complex tasks requiring non-actuating organ system and room for payloads. So in this image, you can see how they developed the organism in vivo. So in fact, first of all, they inhibited the formation of multiciliate cells, and they track the cells with a marker And after that, they took layers of ectodermal tissue stacked on top of each other, and the center of which the layers of cardiomyocyte were placed, as you can see in the second image. And at this point, they let the tissue develop for 2 days and subsequently modified the organism according to the in silico design, as you can see here. Okay, these are live images that show the stratification of the 2 tissue type in the first figures. And finally, we can see a microcautery device that is observed, which is used in conjunction with a surgical forceps, but a forceps that is not shown in this case. They are used to give a shape similar to the in silico design of the organisms. Okay, this is a brief video that shows the building process of the generate organisms. Okay, here you can see the microinjection of the fertilized xenopus embryos with that, and now we can see also that the vitelline membrane is removed from each embryo with the microsurgery forceps in this image. And okay, here is another embryo which is removed the vitelline membrane, and so then the next step is to remove the animal cap of each embryo, and from the same animal cap, the ectodermal layer is manually separated from the inner cell layers that you can see here. And in another step, subsequently, you can see after this process that the remaining tissue is agitated to further aid with the dissociation with the micropipette here. And in this case, thousands of cells are transferred to agarose wells containing normal standard medium, and to promote cell reaggregation in this case, as you can see in this part. And in another step, we can see the cell aggregate undergo compaction, and they readhere, and for 2 additional days. And here you can see a microcautery device that is utilized to define the raw shape of the transferred design. And you can see also a microsurgery forceps that are used to sculpt the final features. And this is the final organism that we can see. Okay, so they want also to test the organism for different behavior. And principally, they tested the final organism first of all for the locomotion in this case. And as you can see in these images, they predict the behavior in the space in silico in this case, and they mimic the locomotion, putting the organism in aqueous environment. So in fact, they wait until it touches the floor, and they measure the shift during 6 seconds in this case, as you can see in this image. So in this case, it's the same experiment in vivo, and in this case, they're measuring the shift during 30 seconds. In fact, okay, so in this case, in this image, you can see that they repeat the measurement, also turning upside down the organism to better understand if the locomotion behavior was not casual and it was due to the contractile part. In fact, as you can see in the second image, they note that when the organism is on their back, it can move in this case. Okay, another important point is that they also want to test the ability of object manipulation, In fact, they put the design in the crawled surface that is shown in these images, and they note that when the environment is strewn with particular matter, motile design spontaneously aggregated the external object, and this behavior is observed both in silico and in vivo. In fact, this is a representative image of in vivo behavior, and as you can see here, the in vivo experiment in fact show an emergent behavior of debris aggregation by an individual within the environment in the first images, and also by a group of reconfigurable organisms over 24 hours periods. So finally, some design involved for displacement reducing hydrodynamic drag via hole throughout the center of the transverse plane. And this more complex topology was realized in vivo, but was not tolerated with the contractile tissue. In simulation, so these emergent features can be expanded as a patch to store and transport object. In fact, in a subsequent round of evolution, patch could be incorporated as a design constraint, and the new goal of maximizing of distance of the carrier object.
Okay, now I will show you briefly a video in which I can summarize what Loredana said before. So, a computer pieced together biological building blocks into different structure. And as you can see here, this is an images of in vitro and in vivo and in silico organisms. So the goal is the movement. Here you can see the building blocks, so the passive one and the contractile one. But most random configuration of the building blocks do not generate useful behavior. So they have to try with different configuration like this, as you can see here. So they try with many different configuration. And with this evolutionary algorithm, they can find a good configuration like this one. As you can see here, it can move slowly, but it can move. Okay, and this configuration was then built from living cells, in particular from the frog cells. And as you can see with the tracker, we can compare the in silico with the in vivo organism. So here this is the contractile part, and here the inert part of the organism. And here you can see a short video in which we can see that in vivo organisms can move like the in silico design. So here is a aerial view, and this is the other experiment that Loredana said before. So to verify that it moves due to design features, the organism was placed on its back, as you can see in the bottom part, and its function was lost. So it can move when it's placed on its back. Okay, they can try with many different configuration, and this process could be repeated indefinitely. So we can have many shape and different combination, as you can see here. For the first application, this is a video in which we can see a collective behavior. So these organisms can move and can round alone, or can touch each other and round together, as you can see here or here or here. And for the object manipulation, they use some debris. And as you can see here, these organisms can move the particles. They also try to transport object, but only in silico, and this is the future shape that they want to try to build. And yeah, unlike the brightness of current technologies, they note that when they cut and produce a damage to the organism, so when this organism is damaged, for example, manually, as you can see here, it can automatically self-repair. So at the end, we can think about a lot of future applications. And yes, of course, I put here only few examples, but for the biomedical settings, an important, very, very important characteristic of this organism is the naturally limited lifespan. So they can live at least for 7 days, more or less 1 week. So, and it is useful for the biomedical settings because they are also they can be degraded, and so they are biocompatible. And for example, here we can see that this organism can be used for a novel vehicle for intelligent drug delivery, for example, or for internal surgery, or for digest toxic or waste products, and moreover for identifying molecules of interest in environments physically inaccessible to robots, for example, for micro robots that are made by different materials, for example, plastic or I don't know, metals, and it could be toxic for the organism. Or at least they can think about the use of this organism for the removing plaque from artery walls, or identifying cancer, or setting down to differentiate or control events in location of disease. For the research fields, yeah, we can imagine also a lot of application, and for example, they can serve as a unique model system facilitating work in the evolution of multicellularity or exobiology, artificial life, basal cognition, and regenerative medicines. For example, another interesting thing that I found is that in the environmental fields, they can also be used in future for cleaning the oceans from microplastics. That is an important thing that of our period. And yes, I hope that this presentation was clear, but if you have questions, you can ask us by the chat. Thank you.
Hello, everybody. I'm Giorgia. Thank you, Michela and Loredana. Thank you all for the attention. So if you have any question, we will be happy to try to give an answer. While I just want to say that we have chosen this work and analyze this work because it seems very interesting to us, especially for its huge number of potential future applications, as Michela say, but all of them still being tested, and many studies will have to be done. So we don't know for sure that all of these can actually be realized, or if it will really work in the future. So now Michela will try to answer your question. So okay, Joanna, say you show them making the model organism using cells from an embryo of Xenopus living, but have they used any other traditional model organism like zebrafish or mice?
Okay, Yona, this is a very good question. And yeah, I asked myself the two questions, but this study is very, very naive, and this is the first time that scientists made an reconfigurable organism like this. Yeah, they didn't use other transitional model organism. They use only the Xenopus organism, but I think that in the future they will try to use different cells. I think, for example, also for human application and like something like induced pluripotent stem cells. I don't know. I think about something like this for the medical fields. For the research, of course, some other organisms could be used, and yeah, for now they didn't try nothing different from Xenopus levis.
Okay, thank you, Michela. Do you have more information about the smart drug delivery? For example, which kind of drug can be suitable, or which type of organ tissue can be targeted?
Okay, no. As I said before, and Loredana too, they perform only a simulation in silico for the drug delivery, and they didn't try to use it in vivo. And what I think is that yes, it could be think about some target using different type of cells, because when they use these two building blocks, they use 50% of inert blocks, inert cells, in this case epidermal cells, also to be improved, to be changed by other different cells. For example, I don't know, cells that can have some receptor, something that can guide it to some specific organs or tissue, of course. But for now, what they did is only in silico, so they don't perform nothing about drug delivery for now.
Okay, Fabiana say, but there are a limit to the lifespan of these organisms.
Okay, allora, they said, they said that they can live for 7 days, and after these 7 days, they stop functioning. And this is also an important thing for the safety of these organisms. But they can also improve and increase their lifespan when they put this organism in nutrient-rich cell culture media. So the lifespan can be increased to weeks or months. So yes, for this point, what they said is that. So okay.
Charlotte say, at the end of the lifespan of the artificial created organism, is the removal of the organism a problem, or does the body eliminate it easily?
Okay, they didn't inject or put this organism into a body, into an organism, a different organism. But what they said is that these organisms are biocompatible and biodegradable. So what I think is that if you made it by your own cells, for example, it can be eliminated easily by the body, but they didn't do it for now, because as I told you, is a very, very naive study. So maybe in the future they can answer to your question.
Okay. Professor Teti asks, could the model be used to test drugs for human disease, or is too far from our species?
Okay, I hope that this model could be used in the future for this for this aim, for this goal. But for now, they perform only this organism with these frog cells. But yeah, of course, if we use different cell type, autologous, for example, cell types, or as I said, for example, induced pluripotent stem cell, or something like this. Yeah, it could be used for the human, but for now they didn't say anything about it because they didn't perform experiment for human or mouse or mammalian organism. So it could be improved to answer your question.
Okay. Ilaria say, what an interesting article. Thank you, Ilaria. Are the reference to possible problems or negative effects?
Yeah, I can show you something. Yeah, I found we found another article that is here, Living Robots, Ethical Question about Xenobot. And if you want, you can read it. So yeah, in this article, they propose some reflection on ethical consideration and something that it could be negative. And for example, they talk about the evolving beyond our control. So the weaponized organism, if they can become sentient, or that if they can acquire reproductive ability. So for now, they can't reproduce, and it could be a good thing because it could be more safe. But in the future, if you, for example, use different type of cells, or for example, neurons or something like this, they can be becoming, for example, sentient and something like this. But for these problems, for this negative thing, they answer with the with this synthesis. So this technique should inform any future guidelines or regulation for the development and use of this biobot. So that is like a Xenobot for the frog, biobot. And yeah, it could be regulated for sure, because also in an ethical point of view, it could be a not point, I think.
Also, Iona asks, what are ethical issues surrounding the generation of these organisms?
Okay, I think it is more or less the same answer. So yeah, they talk about different different ethical issues in this article. Yeah, for example, they think that these organisms could be weaponized. For example, they can be create harmful organisms in the future. But what I believe is that as this technology matures, a regulation of its use and, more important, its misuse should be a high priority for the scientists.
Okay. Fabiana asks, the creation of these organoids seems very complex. Do you think this could be difficult, expensive to achieve on a large scale?
Okay, so they didn't talk about the cost of this technology. For the first part, I think yeah, I don't know if this algorithm is expensive or to use, but I think that they want to improve this technology to become more automatical, and I think the problem is that it's hard to make it manually. They need a very difficult skill to produce, to manually modify this organism, the shape of this organism. So I don't know if it is expensive, but personally I think that it is very difficult to create. Yeah, for the human, for out manually, I mean.
Ilaria asks, is it possible to eliminate genes or silence them? Can these models be similar to know-hows?
Okay, they knock down the a part of the gene that regulate the cilia of the organism. So before layering these tissues, they inhibit the cilia into the embryo cells. And they made it because they want to decrease the how can say the differentiation in the motility, the different motility of the cells. So they want that the movement was regulated only by the cardiomyocytes, and so they knock down these genes. So they perform a sort of knockdown, but yeah, they perform only this. So I think that this model could be used to also to it is possible to eliminate genes or silence these genes into the cells, as all the biology scientists known, but they perform only the this type of editing for now.
Okay. If there are no more questions, okay, I think no more questions. So
Okay.
Thank you, everybody, for the attention.
Yeah, I hope we was all the things that we said was clear, and thank you for the Journal Club team for this, and okay, thank you all, and see you next Tuesday.
Bye.
Thank you. Bye bye.